Hey everyone, and welcome to another video here at Whiteboard Doctor. Thanks for joining us today. Quick disclaimer before we move on. None of these videos are intended to be acted upon as medical advice. Please pause the video here and read the disclaimer in its entirety before moving on. Channel plug. Here at Whiteboard Doctor, our mission is to bring you interesting, relevant, and understandable medical education for all types of lifelong learners, trainees, and practitioners. If you want to follow along, we do have a lovely subscribe button in the bottom right-hand corner of all the videos. Don't forget to hit that like button. And lastly, if you'd like to support us outside of viewing our videos, we have several ways in which you can do that, linked in the video description and pinned comment. Stay well, keep learning, and back to the video. All right, and welcome. It is, let's see, about 8:20 p.m. local time, at least at the time of this recording. We hope everyone is well. Today we're going to mutate a little from our typical coverage. Another pun there, that's 2 videos in a row. Anyone who saw the previous video, y'all get the repeat pun joke there. But we're going to do something, I guess, a touch different. We've done these videos before, but this is going to be a video that's going to talk about how viruses mutate. We actually covered this in a video, at least to some degree. This is our Whiteboard Doctor homepage. We're just going to search Whiteboard Doctor viral mutation on YouTube. And let's see, way back here, for those of you who've been following the channel for a while, you might recognize this very old thumbnail. And this was COVID-19 mutations. Can SARS-CoV-2 mutate into something worse, back in March of 2020. Worse is a relative term, but lots of mutations since then. If you guys are interested in kind of checking this video out, definitely give it a search, because it focuses on SARS-CoV-2, and it was actually before we, had seen many of the mutations that we are now aware of. So we wanted to revisit it. At first we were like, oh, we'll repost that old video as kind of a, relevant, but we wanted to make a cleaner video. Back then, our development skills were a little maybe underdeveloped. So first video back on March 2020, COVID-19 mutations, can SARS-CoV-2 mutate into something worse? And as we just mentioned, worse is a relative term, but it certainly has had plenty of mutations. So in this video, we're going to go over how viruses mutate. We're going to explain it clearly, even though these may look confusing. And then we're going to talk about different viruses, which ones mutate a lot, and how much does SARS-CoV-2 mutate. All right, so with no further ado, we will dive right in. It probably won't be a tremendously long video, but still one that's very important. So little background, right? SARS-CoV-2, the virus that causes COVID-19, has mutated up to, zillion times at this point. We have variants and subvariants and sub-subvariants, and as such, this pandemic has been dragged out now for, going on, gosh, 3 years at this point, a little over that probably. So it's really important to understand how viruses mutate, why they mutate, how often they mutate. We're starting to talk about this a little bit with monkeypox. That was the previous video we put out, was on this new virus that causes this disease called monkeypox. And is that virus mutating? We said less likely, given the type of virus it was. So you'll understand all that at the end of this video. So a little bit of background. There's different kinds of viruses, right? And the different kinds depend on a lot of things, but one of those things is the type of genetic material the virus carries. So just like humans, our genetic material is DNA, deoxyribonucleic acid, and that is the building blocks of life, right? That is the code that is then turned into proteins, and proteins are what build us. Well, viruses have genetic material as well, but their genetic material can be packaged in a few different ways. Sometimes it's a single strand of ribonucleic acid. All right, that's RNA. In humans, typically DNA is our genetic material that's then turned into RNA, and then RNA is turned into proteins. And proteins are what actually kind of build us. Well, some viruses skip this first step and they're just RNA. All right, there's also double-stranded RNA viruses. So instead of a single strand, they have 2 strands of RNA. Rare, but possible. And then there's double-stranded DNA viruses. All right, there's actually single-stranded DNA viruses as well, but these are rare. So single-stranded RNA viruses are the very well-known now SARS-CoV-2, right? The virus that causes COVID-19. This is a single-stranded RNA virus. Monkeypox, the virus we just talked about in our last video, is a double-stranded DNA virus. And the reason this is important is it is related to how and why these viruses mutate. So here's a diagram that we're going to go through. All right, the red is a human cell. And this is kind of step one. This is the virus, right? Circle, the blue in the middle is a single strand of RNA. Well, that virus attaches to the human cell, and then it is often sometimes endocytosed. Endocytosis, which we could have drawn that step, but endocytosis is where part of the human cell membrane kind of buds off, and the virus sits in the inside of that before breaking out. Sometimes the virus actually kind of molds into the cell wall, and in either step, it's going to dump its genetic material into the human cell, right? And that's what we see here in step 2. This is a single strand of viral RNA, viral genetic material. And a few things happen then. The first thing is that this viral genetic material finds what's called a ribosome. And this is a human ribosome, and what a ribosome does is it takes RNA and makes it into protein, right? And our protein is what builds us, our structures. So what we see here is this ribosome grabs this viral RNA and it does two things. One, it forms it into something called the RNA-dependent RNA polymerase. What this essentially means, polymerase, ACE at the end means it's an enzyme, and it's an RNA dependent, so it's an enzyme that is dedicated to RNA polymerase. So it takes RNA and it creates more RNA. All right, so this ribosome grabs some of the viral RNA that code for this RNA dependent RNA polymerase, and it creates it, right? So this ribosome is reading this strand of viral RNA, and this viral RNA is coding this RNA dependent RNA polymerase, and that's what's created. The other thing the ribosome does is it's reading all of the different viral RNA. And some of that viral RNA is coded just for viral proteins, right? The building block, the structure of the virus. So in addition to making the RNA-dependent RNA polymerase, it also makes all these viral proteins. This step here is, we're going to call that 4, this will be 3, this will be 3.5. This step here is where we start talking about mutations. Because what this RNA-dependent RNA polymerase is meant to do is it's meant to replicate the RNA, right? It's RNA-dependent, so it takes RNA, and it's an RNA polymerase, so it makes another pair of RNA. So this RNA-dependent RNA polymerase goes and grabs some of the viral RNA and replicates it. It duplicates it. So it kind of reads the RNA, and then it kicks out a second strand, right? So it's reading one strand and kicking out another strand. And the thing with the RNA-dependent RNA polymerase is it is imperfect. It makes mistakes, right? It's building this genetic sequence that's supposed to be identical to the sequence it's replicating, but it makes errors sometimes. Just like if you were typing out a document, you were looking at it, on the side, and you were typing it on your computer, you would eventually start to have a few typos. Now, if you're really good at typing, there'd be very, very few typos. If you were bad at typing, there would be a lot of typos. Secondarily, if there were typos and you were paying attention, you could fix those typos. If you weren't paying attention or you were unable to pay attention, you could not fix those typos. And we'll get into some of those analogies in a little bit, but essentially RNA viruses, they don't have the capacity to fix their typos, and they type very, very quickly. So they make errors and they don't fix them. And that is a mutation. It is a random error made by this RNA-dependent RNA polymerase. All right, so if we scroll to the side just to kind of spell it out a little bit more, let's just draw kind of what we'll say is an RNA, right? So these are different nucleotides or nucleosides, depending on what you're talking about, that are making up a strand of RNA. Okay, the different colors represent different ones. And this RNA-dependent RNA polymerase, which we'll kind of just draw here as two circles, this is one RNA-dependent RNA polymerase. And what it is supposed to be doing is it's supposed to be identically replicating this strand of viral RNA. But it's imperfect. So as it's going down, it accidentally might sub in, instead of a black nucleoside, it subs in a red one. And then it just, ooh, apologies. Then it just keeps going, right? And it finishes the sequence, and it kicks both of these out to be packaged into new virions as if they're identical, but they're not. There's a mutation here. And RNA viruses don't really have the capacity to fix that in any meaningful way. So you get an RNA sequence that has a mutation in it, and it's totally random. Rather, this is red, or it could be blue, or it could be green. It is just a random mistake that it made. All right, so what happens then is in this single infection here, the RNA-dependent RNA polymerase replicates this RNA a whole bunch, but it makes a mistake, right? And you get a mutated strand of RNA. Well, that ribosome, right, the human ribosome making the RNA into protein, makes a bunch of viral proteins. They form a new virion. It's called an individual viruses, a virion, V-I-R-I-O-N, a virion. And that virion is then packaged with the different genetic sequences, right? They go into the virions, but one of those genetic sequences, the blue on black thing maybe is a little confusing, but one of those genetic sequences here has a mutation in it. That doesn't affect this first group of viruses, right? They all have the normal protein structure of the virus that infected the cell initially, which you can see here. But what happens when this mutated virus, I shouldn't say that, this virus that has a genetic sequence that has a mutation in it, what happens when that infects the next human cell? And that's what you can see here, right? Is you have this mutated strand of RNA. There was a random mistake, which is right here. You get the human ribosome, as we talked about, and the human ribosome turns this RNA strand into proteins, viral proteins. But unlike here, where you can see the viral proteins were black, this mutation codes for a different protein, a random different protein that is kind of demonstrated here in the yellow, right? And if this is a functional protein, the viral structure is still going to be assembled. This virion is just going to have a mutated protein. Sometimes, though, this protein is not functional. And if it's not functional, then you can't actually form this new virion, and this mutation just dies out, right? Because it can't form functional virus. But if it is functional, and that does not necessarily mean it provides a benefit. It might even provide somewhat of a detriment, but as long as that viral protein is functional, it'll form this new virion, which is now a mutated virus, right? You can see the yellow protein sticking off the surface, whereas the original wild type or native strain had a black protein off the surface. So now you have this new variant. And this variant has a mutation that led to a mutation in its RNA code that led to a different protein, right? Because the RNA codes for the proteins. So this mutation said, hey, instead of putting this amino acid base, which amino acids build proteins, it said instead of putting the one you usually do, we're going to put this different amino acid in the sequence, and that can create a new protein. The thing to keep in mind, though, the first lesson is that these mutations are random. The second lesson is that they could be beneficial to the virus. And I put beneficial in quotes because the virus isn't thinking. It's just a random mutation that might randomly benefit the virus. It also could be detrimental. This could be a non-functional protein that the RNA mutation led to, and that might just die out. Or it might be neutral. It might not provide a benefit or a detriment. It might just be a random neutral mutation. Many times even the mutation doesn't change the overall viral protein structure. So sometimes you will even have, I'm going to erase some of this just so it's easier to see. Sometimes you'll even have mutation in the genetic sequence, but it doesn't even really change actually the viral proteins being made. It's just a mutation that's so kind of meaningless that the protein's the same. Or sometimes, as we said, It might be a mutation that leads to a non-functional protein. Let's say these are kind of torqued off like this, and these torqued off proteins are detrimental. They can't infect cells, and then that just dies off. All right, so it's a random mutation, and that random mutation many times could be detrimental and kill the virus, or neutral and not do anything at all, or not even change the viral protein. And every once in a while, the virus gets lucky and the random mutation just happens to be beneficial. So what does this look like then? So with SARS-CoV-2, with COVID-19, we see like all these different variants that start to kind of take hold in a population and become the dominant variant. Well, what does that look like? So the first thing we want to read out is a virus isn't making the decision to change its genetic sequence to be this, that, or the other, right? It's random. But if the virus could choose, the most optimal mutation would be for the virus to be highly transmissible for the longest period of time. And if you think about what that phrase means, that essentially means that the best thing for the virus, because all the virus, the virus wants to just replicate as much as it can. It's not alive, it's not making decisions, but that is what it does. It kind of perpetuates its genetic sequence. And a mild illness that sticks around for a long period of time and is highly transmissible will allow the virus to mutate the most. Whereas if it's more lethal and it just kills the person quickly, it might not transmit to many people, right? Or if it's less transmissible, it's also not going to transmit to many people. So what we drew out here was kind of a diagram of that. This is the old, right, with all the black, kind of the wild type virus. And we're going to say that this wild type virus, one person typically infects 2 more people. Here's 3 of the mutated viruses. One of them is more transmissible. So this is the old one. One of them is more transmissible. So instead of one person infecting 2 people, one person can infect 3 people. Another one is more lethal. So it still infects 2 people. One person still infects 2 people, but one of those people dies. And then the third is less transmissible. One person only infects 1 person. So what does that look like then? What we drew out here was 1, 2, 3 kind of infectious cycles. So we could show you what happens. So first infectious cycle, which is going to be right here, right? This is first infectious cycle. What you see is that the more transmissible virus, the one person infects 3 people. The more lethal virus infects 2 people, but one of them died. So that just leaves 1 left to transmit the virus. The less transmissible virus infects just 1 person, right? And the old virus infects 2 people like we talked about. And this might be like, we don't really see much difference here. But what happens in the next cycle? Well, each one of these people with the more transmissible variant, the mutated virus, infects 3 people, right? So 3 all of a sudden goes to 9, right, during this second infectious cycle here. And the more lethal virus, there is 1 person left. 1 person infects 2 people, so it goes back to 2 people, but then one of them again dies. So there's just 1 left over. The less transmissible virus, we X'd it out, but technically it'd be, this 1 person infects 1 person, right? And the old virus, 2, goes to 4. So at this point still, during cycle 2, the less transmissible virus is kind of dying out, just barely holding on. The more lethal virus is just barely holding on. The old virus is still kind of holding on, but the new, more transmissible virus is starting to take root. Then we get into kind of infectious cycle 3, right? And now these 9 people, each one infects 1 other person, and that gets to 27 people infected. The more lethal virus still is just sitting at 1. The less transmissible virus is still just sitting at 1, and these at this point are kind of dying out. And the old virus still is infecting some people, but it's starting to become the minority, right? Because the more transmissible virus has become dominant. And that's how we start to see these different variants kind of becoming the dominant strain. Now, there's a lot more to this. There's environmental things to keep in mind. There's kind of population densities and all sorts of stuff, immune escape and all that. But this is kind of a structure that you can think about it. So the most transmissible virus is the one that typically will become the dominant variant. And that does not mean the most lethal, right? All right, so the last part of the video here is just how often do viruses mutate? This is going to be kind of some real world data, which we thought was interesting. So this here is a graph from this article here. It's going to be linked in the video description, Viral Mutation Rates, published in the Journal of Virology. And what they're demonstrating here is on the y-axis, the mutation rate, okay? And this mutation rate is mutations per nucleotide, so per single genetic block. Nucleotides make up the RNA and DNA, per cell infection, so every time it infects a single human cell. And the point we wanted to get across here was that DNA viruses are represented, you can see double-stranded and single-stranded, are represented by these here, all right? These are actually bacteria down here, so bacteria mutate much less than viruses do, which is interesting to know, right? But double-stranded are here, DNA viruses are here, whereas RNA viruses are up here, all right? So RNA viruses mutate much more than DNA viruses do. And that's because the RNA is kind of replicating quickly and not fixing itself. Whereas DNA viruses, the genome is much longer and kind of more complex often. And there are some tools that DNA viruses have to fix any of the random mutations that occur, right? And as an example of this, right, RNA viruses is the virus that causes COVID, SARS-CoV-2. DNA viruses is the virus that causes monkeypox, which again we talked about in that last video. Definitely just go to our page and check that out. This right here is a viroid, it's a bacteriophage, and these down here are bacteria, right? So they replicate much less. The other thing to note is the bigger the genome size, right? So bigger genome sizes go here, the lower the mutation rate. So bigger DNA viruses and bigger RNA viruses mutate less or fix their mutations more often, whereas small RNA viruses are the ones that have the highest mutation rate. And if we want to talk about kind of real world viruses, so this is a table. We're going to go actually back to the study. I think we kept it pulled up. Yep. So this is a table from the study, and it is ranked in order of kind of highest mutation rates. And you can see the mutation rate here. And bacteriophages mutate the most. A bacteriophage is actually a virus that infects bacteria. We actually talked about it. We put out a video on antibiotic resistance and how that's a huge threat to the globe. We'll link that in the video description or in the top right corner. Check that out if you're interested. But then other viruses, tobacco mosaic virus, human rhinovirus, right, that causes kind of upper respiratory infections, is a big one that can mutate quickly. Poliovirus, which we don't see too much of anymore. And then here's some other ones. VSV, measles, these are ones that you don't really need to come across very often. And you can see all these are RNA viruses. Then the DNA viruses start to come in as ones that have slower mutation rates. You're getting kind of 10 to the -8 there. All right, so then the last part of the video is, well, everyone's interested in, SARS-CoV-2 and COVID. Where does that come in? So the SARS-CoV-2 virus, the virus that causes COVID-19, its mutation rate is often quoted at 10 to the -6. And what that equates to is 0.00001, and that's mutations per nucleotide per infectious cycle. And when we say nucleotide, what we are referring to is, we're going to scroll up to our picture. This is a strand of RNA, right? Each one of these smaller areas is a nucleotide or a nucleoside. We actually can't remember which one is the correct verbiage if it's RNA. So we're just going to write nucleotide, but each one of these is a nucleotide. So the study here is essentially saying this many 10 to the -6 mutations per nucleotide per infectious cycle. Well, what does that look like? There's this really interesting article published in PNAS, which is the maybe Proceedings of the National Academy of Science. That might not be appropriate, but it's something like that. And we'll link that in the video description too. And what they talked about was the SARS-CoV-2 genome is 30,000 nucleotides long. And they did some math here, and there were some assumptions with this math, so take it for what it's worth, but it does give you kind of an idea. Each person, they said, when you get a SARS-CoV-2 infection, usually at least a few infectious units, which is like individual viral units, infect a person. It's often not just a single infectious unit. And each human cell, when it's infected, gets about 10 to 100 infectious units. And again, a single infectious unit is just a single virus or a single virion. And each person at the peak of their infection with COVID-19 tend to have 3 times 10 to the 5 to 10 to the 8 total infectious units, right? So if we do our math, let's see, 3 times 10 to the 5 is going to be 5 zeros. 1, 2, 3, 4, 5. So that's 300,000 infectious virions to 10 to the 8, which is 8 zeros. 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3. So to 300 million infectious units. And these are single virions, right? And a virion, as we said, is just a single virus. So what they essentially equated was, with some of these assumptions, which are backed by a lot of the evidence, it equated to that each virus, each individual virion replicates 3 to 7 times per infection. All right? And if we look at the mutation rate up here that we talked about, a single virion, a single virus then, on average has 0.1 to 1 mutation per person infection. So what that means is if you're infected, a single virion, one virus that's replicating inside of you, will have 0.1 to 1 mutations per single virus. They then said that the evidence shows that, right, I can give that virus, that single virion, to someone else that has that 0.1 to 1 mutation, but usually it's a 4 to 5 day delay between infections. And if you count that in and you consider that virus, right, that one virus that infected you and mutated, now it's going to infect someone else in 4 to 5 days, they said that per month there will be about 3 mutations per a single viral unit per month. And that's a viral unit that kind of survives and keeps infecting people. So if you think about, oh, not sure why that popped up, apologies. If you think about how many people have been infected and how many viruses are around, there's, a ton of mutations that SARS-CoV-2 has gone under over the past 3 years. And that's why we have all these variants, because remember, the variants are just the mutations that have somehow randomly provided a benefit to the virus so that those variants have taken hold. There's been up-teen zillion mutations that, 1, haven't done anything, or 2, have been detrimental to the virus, or 3 were just kind of neutral and never really led for that variant to really take hold. So we just wanted to revisit that, some interesting stuff. Hopefully this better informs you on kind of how these viruses mutate, why, how often, and what to expect going forward, and maybe provides a little context for understanding all these variants and subvariants and etc. With COVID-19, because inevitably there will be more viruses coming around. So let us know what thoughts, comments, questions you have down below. Definitely hit the subscribe button if you want to follow along. We appreciate you. Comment. Let us know how we're doing. Let us know if there's anything you want us to cover that we haven't. And as always, stay well, keep learning, and we will see y'all next time.